I pretend to be.
I think you are.
So,
I think you're on it.
good evening, everybody. We would like to welcome you to the third webinar that's being sponsored by Chamow Associates, and we're very pleased to have two presenters tonight. The first presenter with us is Darlene, or Dar Rosario, and as I mentioned earlier, before we started recording, she's an experienced regulatory affairs professional and compliance professional. She's got over 30 years of research and development of drugs and biopharmaceuticals. Dar holds her MBA from St. Mary's College. She got her BS in chemistry from Eastern College University and her regulatory affairs certification, RAC. She is also a holder. She's an active member of the Regulatory Affairs Professional Society, or RAP Society. So we welcome you, Dar. We're very pleased to have you join us this evening. And the other presenter is Dr. Stephen Chamow, or Steve, and he has over 30 years experience in the biopharmaceutical product development. He's the founder and president of Chamow Associates, Inc., a consulting group working with the biotechnology companies to design and implement strategies to develop new therapeutic products. Steve was educated at the University of California, UC Santa Cruz, where he got his BA in biology. He got his PhD in biochemistry from UC Davis and completed his postdoctoral training at the National Institutes of Health in Bethesda, Maryland. He is an author or co-author of more than 50 scientific publications, and I think that becomes 51 as of today, based on something that he sent out to the company. And he also holds patents and is a co-editor of 2 books. So without further ado, I'm going to turn over this presentation to Steve and Dar.
Thank you very much, Kelly. Hello, everyone. It's a pleasure to be here with you. We'll be talking for the next hour, a little over an hour, about developing therapeutics at an accelerated pace. And I'm going to share my screen here, so give me a minute. All right, great. Well, for those of you who have attended one of our other webinars, this is the third webinar that we've done. And this one is a little bit different. This is geared a little bit more toward some of the details of drug development, monoclonal antibody development, and it's so we're going to dive into the weeds a little bit more on how drug development is done, and specifically aspects of the process that can be speeded up a little bit. So with that, I'd like to move to the, just tell you a little bit about what we're going to be talking about today. So we'll start off with potential sources of drugs for COVID-19. We'll then move to regulation during a pandemic. This is an area that Dar will talk about. And specifically, you may have heard about an EUA or a CTAP. Those are both FDA programs that Dar will introduce you to tonight. Then I'll talk about the SARS-CoV-2 virus itself and a little bit about the biology of infection, since this is important to understand the mechanism of action of monoclonal antibodies. Then we'll move into the focus for the evening, and that's on how to accelerate monoclonal antibody development. We'll introduce that with pre-pandemic timelines and talk about how those timelines are conducted, and then what opportunities there might be for faster timelines to make monoclonal antibodies in a shorter period of time. And then we'll close with showing you some examples of how these accelerated timelines are being applied and have been applied over the last several months with leading new therapeutics from 5 different companies that are currently anti-SARS-CoV-2 monoclonal programs that are in the clinic. Before we get started, I want to mention, for those of you who might have noticed this when you registered, we have a number of pre-reads that we are recommending. I would suggest, if you haven't had a chance to take a look at these, we have links on our website, and please do. They're delightful reading and very interesting. The John Cohen article from this month's Science about antibodies that may curb the pandemic before vaccines is a good overview of what we're going to talk about, the leading antibody programs we're going to talk about tonight. The second article goes to the heart of our presentation and discussion, and that's Brian Kelly's article that was published in May in Nature Biotechnology that's developing therapeutic monoclonal antibodies in pandemic pace. And then the third article that we suggest you might want to read is a delightful article by Paul Tullis that appeared in the July 20th issue of the New Yorker. How a potential treatment for the coronavirus turned up in a scientist's freezer. So let's start off by reviewing what are the potential sources of drugs to treat COVID-19. They fall into 3 categories. We have repurposed therapeutics, convalescent therapies, and new drugs, which are new chemical entities. So if we look at repurposed therapeutics, what are these? They are existing and marketed products for another clinical indication or a disease. So these are pre-existing drugs, and they're being repurposed for this particular indication. So the special regulatory pathway for bringing these types of products to this disease indication is the EUA, and again, you'll learn more about that in a minute. Secondly, convalescent therapies. These are processed blood from recovered COVID-19 patients. There's some very exciting news just over the weekend about convalescent therapy that we'll tell you about. Also, the pathway there is an EUA. And then for new drugs, these are newly developed and never before tested products. These are not appropriate for EUA. Rather, the program under which these are brought forward is called the CTAP or CTAP. All right, so to cover regulation during a pandemic, I would like to introduce my colleague, Dar Rosario.
All right. Thanks, Steve. Thanks for the invite. So what I want to do in the next about 8 slides is give you a preview on some of the legislation and guidance that have been issued or have been around to help deal with pandemics, bioterrorism, emergency threat situations like that. When Steve asked me initially if I wanted to participate in this talk, and I started looking at what the technical aspects he was going to present, I really didn't think it was going to add much value to the talk. For full transparency, I'll tell you that I'm not working on a COVID drug. I'm not working on a COVID monoclonal antibody or a vaccine program. It would be nice if I were, only because I would have more of a hands-on approach to this talk. But in doing the preparative work for this, it's very clear to me that a lot of the legislation, in particular the guidance and some of the initiatives from FDA around this, mimic what we've seen before in terms of things that have gone on in the world. For example, the HIV problems, the bioterrorism problem we had in 2002. And with every issue, we know that more regulations come out, more guidance come out, more focus comes out in a particular situation. And that's the case in this particular situation. So I really appreciate him asking me to do this. And I appreciate the learnings that I've learned, and I want to share them with you. So I'm by no way going to do a regulatory 101. I would bore you to tears at 5:20 in the afternoon. But what we want to do is point out a couple of what's going on in terms of initiatives that are helping to align this talk today in terms of accelerated pace of monoclonal antibodies. Because of course, if you accelerate drug development, the regulations have to keep up, and in fact, they have. So this particular slide right here just lists the related legislations with regard to medical countermeasures. And I noticed that Steve has stayed away from defining the acronym EUA, and you can see this is the Emergency Use Authorization. And what I've done here is really list a number of the legislations that focus on this EUA, the Emergency Use Authorization, and how it's kind of evolved and how it's kind of gained ground, leading to the FDA being able to authorize products for distribution. Of course, you all probably can think back in 2001 on September 9th, and you probably know where you were. And as a result of that, the Bioterrorism Act was created. And the focus was to strengthen the nation's ability to respond to public health emergencies. It made MCMs, medical countermeasures, a priority. And you can see here that there were some other initiatives as a result of that, but really the focus of prioritizations of medical countermeasures. In 2004, we had the Project BioShield Act, and this is where the FDA commissioner was giving authority to issue EUAs. And then the act, which is clearly defined in the FD&C Act sections 564, 564A and B. So if you're ever bored to tears or really that interested, if you read all the way back to the act, you can see how these are linked to this initiative, the EUA. The Pandemic and All Hazards Preparedness Act of 2006 was an amendment to the act, and it established in the Health and Human Services a new secretary to handle preparedness and response initiatives. That act was then reauthorized, and more provisions were further given to the FDA's mission to develop and make MCMs available. We have an act in 2016, and as a result of that act, there was a guidance, what I'll talk about on the next slide, which is specific to the EUA and how FDA is authorized to implement that. And then in 2019, the Pandemic and All Hazards Preparedness and Advancing Innovation Act was a reauthorization of several of these acts, further clarification. So you can see since 2002, there's been a big focus on making sure that the nation's ready for bioterrorism, acts with regards to agents such as biologics and radiation and nuclear weapons, and specifically pandemics or threats to the United States. So next slide. So these legislations, as most of you know, if you're in drug development, lead to guidance. Guidance is how the FDA clarifies and implements these legislation. And the EUA authorization is clearly delineated in this 2017 guidance. It replaced the 2016 guidance and some others that were available. But really, this particular guidance explains the FDA's current thinking on authorization. It provides general recommendations and the process by which one can request an EUA, and I'll go over that a little bit in the next slide. And I said earlier, these were reauthorization or expansion of the Food and Drug Act under these sections. It gave them legal authority to strengthen and get us ready for public health and domestic emergencies. And it authorizes the emergency preparedness and making sure that the medical countermeasures, which include antivirals, biologics, vaccine products, blood products, therapeutics, and devices. Next slide. So on the next slide, I wanted to talk about the EUA a little bit, but I think it's really important to understand what the EUA is not. The EUA is not an approval. It's an authorization under this emergency situation. for the FDA to authorize products to be introduced, medical products, into interstate commerce, only intended for use under this or during this actual potential emergency. So yes, when the emergency is undeclared or is withdrawn, these products will have to be withdrawn. And that's because these candidates are medical products where their uses are not approved or cleared or licensed, or it's an unapproved use of an approved medical product where they found evidence of effectiveness or may be effective and a label or a fact sheet with issues to help distribute this product in use. The FDA looks at 4 statutory requirements with regard to issuing an emergency use authorization. Of course, it has to be a serious and life-threatening disease. And the most interesting of these is the evidence of effectiveness has been reduced. So where the evidence of effectiveness is different under the regulatory process or the regular statutory process for approved drugs, this bar is lowered. So the evidence is that it may be effective. And you can imagine that that's important because if you were going to generate the kind of data we need right now for regular approvals, it's a lot of data, a lot of patients, a lot of trials. Also included is the risk-benefit analysis. So FDA will conduct a risk-benefit analysis of the evidence and issue that EUA under that premise. And of course, no alternatives exist. It is separate and distinct from the use of medical products under an investigation or an IDE or an IND or an investigational drug or an investigational device exemption. In this circumstance, an IND or an IDE is not required for potential EUA products. And part of the justification of that is because the FDA expects that a number of these requests are going to be for late stage or later stage products that may already have an IND open or an IDE open, or maybe products that are approved that are just seeking an indication to support this actual emergency. As always, the FDA has encouraged and is encouraging companies to submit a formal request. They've issued what are called pre-EUA activities, pre-submission activities, where if you think you have something that you might be submitting a request for EUA under, have a conversation with the agency. Next slide, please. So one of the initiatives that FDA then quickly, well, I would say quickly back in May, put into place was this CTAP, this COVID Acceleration Therapy Acceleration Program, CTAP. This is specific for therapeutics only. The FDA has issued an Operation Warp Speed type program for vaccines. They've also issued another initiative called Interact, where, believe it or not, it's an opportunity to have a non-formal interaction with the FDA. It's a one-time event where the FDA will give you some guidance on particulars for something that's very novel, a novel biologic or a novel problem that you may be having. So the FDA is really all hands on deck to put together some initiatives that are focused on getting products, MCMs, very quickly to the patients and people that need it. So this is a special emergency program for coronavirus therapies. Again, as I said, the FDA is trying to use every available method to move these treatments as quickly as possible. And FDA has issued key guidance on these. They're listed here. They're very clearly defined in the FDA's website. This is akin to a fast track program on steroids where you request a pre-IND meeting very quickly. The process is defined, but FDA is opening their doors and wanting to meet quickly and early with programs that believe they could take advantage of the CTAP program. Next slide. Just want to spend a few minutes, and again, this is on the FDA.gov CTAP website. They update it pretty routinely. This update is as of July 31st. If you didn't know what was going on and trying to keep up with the plethora of news that are out there, you can see that there are 570 development programs in the planning stages. That means pre-IND stages. And this excludes vaccines. I just want to make that clear. There's 270 trials reviewed by the FDA. Back in July, there were 2 EUAs issued. There's still 2, but they took away the chloroquinone and am I saying that right? I'm tongue twisted.
Hydroxychloroquine.
Thank you. And nothing, we all know nothing's been approved. Thanks, Steve. Next slide. Let me take a sip of water. This slide just breaks down the types of treatments. You can see there's single agents and combinations. They're all over the map: antivirals, gene therapies, immune modulators, neutralizing antibodies. So there's a lot of studies being initiated. And if you want to go to clinicaltrials.gov, you can get specifics around these if they have indeed filed in that portal. Next slide. Here you can see this, what's happening in the stages by the stage of COVID trials in the US. You can see a number of late stage. Some of these could, as soon as data are available, make a request for EUA. They could also continue and seek approval specifically under the COVID program. And then you can see that there's 60 in early trials that are evaluating safety and dosing, and the later ones, of course, are looking at efficacy and safety. So if you've got time and are interested, the FDA website has plenty of information on what's going on. Next slide. I think this is where oh, there's one more. So Steve talked about earlier about repurposing therapeutics, and we wanted to show 3 examples. Many of you may be aware of remdesivir. It's an old, well, 2014 antiviral drug that was developed to treat Ebola and first approved in 2014. And it does show efficacy in the SARS-CoV-2 in vitro data. So they fairly quickly put a package together, and you can see that they were authorized by FDA under EUA on the 1st of May to distribute product for this specific use, hospitalized patients. These patients were severe, and it reduced that time that people were hospitalized by 4 days. Recently, I just read that Gilead announced that they had filed an application for actual approval of this in severe patients and moderate severe patients. So that's good news. Dexamethasone, I'm sure a number of you have heard this drug, is a corticosteroid first approved in 1961. Researchers in Oxford found that it's very effective in treating, again, severe cases. That trial is ongoing. This is for patients on ventilators, and it reduces the risk of death. They are authorized in the UK to do this. We haven't seen an EUA status here in the US. We don't know if there's a plan to submit that. We all know that there's many manufacturers of dexamethasone, but we'll stay tuned for that one. And then just recently, a convalescent plasma, a program that was initiated by the government, FDA, in collaboration with medical centers and Mayo Clinic. We're evaluating blood, plasma from recovered COVID patients. They recently, on the 23rd, got an EUA that was based on previous human experience, some animal models in hamsters and mice. They had some safety and efficacy data from an expanded access program that Mayo Clinic was conducting clinical trials under, and certainly a risk-benefit assessment. So these are 3 examples of repurposed drugs that are currently being used for COVID. Steve, I'll turn it back over to you then.
All right. Well, great. Thanks, Dar. So let's shift our attention now to the virus itself. The virus that causes COVID-19 we refer to as SARS-CoV-2. This is a schematic structure of the virus. This image is quite familiar to all of us. We see this image, the one on the left anyway, almost daily as we go through our lives. And I want to explain a little bit about what these different elements of the virus are. But before I do that, I want to point out that viruses in general, and this virus in particular, are really very small. How small is this? Well, SARS-CoV-2 is 120 nanometers in diameter. And so what that means is that if you take a head of a pin and line up SARS-CoV-2 viruses across the head of that pin, in other words, the diameter across the diameter of the pin, you could fit 2,000 of these viruses on the head of a pin. So that's how small these guys are. The surface of the virus is shown on the left, and the red structure is the envelope, the viral envelope, and all of the colored projections coming out of the envelope are all proteins of various types. They're all viral proteins encoded by the viral genome. So if we take a slice through the virus, then we get the image on the right. And here again, you see the envelope in red, and then the variety of proteins embedded in that envelope. And I'll go through those quickly. The purple protein is called the spike glycoprotein. This is a very important glycoprotein to the virus. It's the primary means by which the virus communicates with the outside world. And we'll come back to that in just a minute. The green protein is called the M protein. Then the turquoise protein there is the hemagglutinin esterase dimer. And the yellow protein is the E protein. And all four of these viral proteins have distinct functions that the virus uses as part of its life cycle. In addition is the nuclear material, or the nucleocapsid. That's the circular coiled structure in yellow. This is located inside the envelope. This contains a viral RNA condensed with N protein, which is another viral protein. So going back to the spike glycoprotein, or the S protein, I mentioned that this is how the virus communicates with the outside world. It turns out that the spike glycoprotein has evolved a very specific lock and key mechanism of binding to a particular protein that naturally occurs on the surface of human lung cells. And we'll learn about that in just a minute. But it's that lock and key interaction that allows the virus to bind to lung cells and to ultimately infect those cells. This spike glycoprotein is an important player in the viral life cycle. So in the next few slides, I'm going to just schematically illustrate what happens when the virus infects a human lung cell. So this is panel 1A over here on the left is a schematic image of a virus. And you can see the hairpin, the black hairpin structure is the spike glycoprotein. A human lung cell is schematically shown in panel 1B, and this is the cell prior to any encounter with virus. So I just want to very quickly go through, remind you of the biology or the structure of the cell. Cells are membrane bound, so there's a cell membrane, and that's shown in white. And then the inside of the cell is called the cytoplasm. And then inside that are a series of organelles, one of which is called the ribosome, and that's where the protein making machinery is. Then there's an organelle known as the endoplasmic reticulum that is involved with the ribosomes in the assembly and secretion of proteins. What's shown in blue here is a structure embedded in the cell membrane, and that is a protein called ACE2, or angiotensin converting enzyme 2. This is the protein that is the receptor for SARS-CoV-2. So when the virus approaches the human lung cell, and you look at panel 2, and you'll see what happens here, the virus recognizes and binds to the ACE2 receptors on the surface of the cell. That's a specific lock and key interaction that allows the virus to then prepare the cell for infection. Infection occurs here in panel 3, where bound virus enters the cell. It's then uncoated, and the genetic material, or the nucleocapsid, enters the cytoplasm. The viral RNA attaches to the cell's ribosome and is translated to produce viral proteins. So in this way, the virus is beginning the process of hijacking the cell's protein-making machinery taking over the cell for its own purposes to make viral proteins and ultimately viral particles. And you can see that process proceeds from panel 3 to panel 4, where the viral polymerase is the first protein that's made, viral protein that's made, and then that allows for additional viral proteins to be produced. New virus particles are produced inside the host cell. You can see that new virus particle within the endoplasmic reticulum. That's where the particles are assembled. And then from the endoplasmic reticulum, virus particles are ejected out into the extracellular space, and they go on to infect new lung cells. So I've shown you here the life cycle of SARS-CoV-2. A similar process occurs with many viruses, but it's important to understand that this is an amplification mechanism, so that what I've shown you is one virus infects one cell, which results in the production of one virus. And that's a simplified story. In fact, it's several viruses infecting a cell, and then that cell is completely overwhelmed by the viruses, and all it does is produce many viruses as a result of that infection. So what we're trying to do is we're trying to interfere with one molecular event, and that's the binding event, that initial interaction of virus with ACE2 receptor. And the goal of an anti-SARS-CoV-2 monoclonal antibody is for it to interfere with the virus's ability to bind to lung cells to prevent this infection. So we raise antibodies, or we create these molecular specific drugs that bind to the spike glycoprotein of SARS-CoV-2, and in doing that, they prevent ACE2 from recognizing and binding to the viral receptor. So it's kind of it's jamming up the works of the lock so that the key can no longer fit inside. That's the whole point of these kinds of drugs. The better the drug, the more completely we gum up the lock so that the key can't work. So with that then, brief understanding of how these kinds of products might work in the biology of the virus, let's move to accelerating the monoclonal antibody development itself. Dar?
Yeah, so on this slide, this talks about the stages. This slide specifically says monoclonal antibody product development, but if you've been in industry long enough and working through the drug development continuum, this should be familiar with you. But we want to focus in a little bit on the monoclonal antibody identification and the current process for that. If you're not familiar with this, what happens is that a new candidate is discovered, or a potential drug candidate is discovered in discovery and research, moves into the laboratory testing, where we prove that it works in an isolated virus. Additional tests go on there to further characterize the molecules. And at the end of this point, we have a lead monoclonal antibody, in this case, identified. That lead candidate, or that lead monoclonal antibody, is moved into animal testing, where it's evaluated in vitro and in vivo to generate efficacy, a toxicity profile that's acceptable. And you can see right at this point, you start to see the manufacture of the drug initiate and begin and move. All of this analysis and all of this work, of course, is targeted to file, in the case of this particular case, the investigational drug, the IND, so that we can begin clinical testing in humans as quickly as possible. I don't know how long many of you have been around, or how long you've been in drug development, but if you recall, one of the paradigms of speedy drug delivery I think this is kind of that element, and even speedier, especially now because of the need. But down here, you can see that at the beginning of the manufacture and through the manufacture of the drug before 2017, this took anywhere from 18 to 21 months. Since then, we've been able to reduce that time to make this product and create the drug and be ready for IND filing in 12 months with the appropriate data, the appropriate specs, methods, manufacturing, those things that we need. And I would also argue that it's probably even sooner because most of us have pre-IND meetings and that information is needed for that. So the question I think we're proposing, and Steve is going to be leading, is how do we accelerate it even more? How do we get it below 12 months? Steve?
So let's take what Dar just said, and that's the current monoclonal antibody development timeline in 12 months, and dissect that a little bit to understand how that works. So we have here a timeline and the various types of tasks that take place as part of this drug development scope are color-coded. So the first stage of work is cell line development, that is coded in yellow. And you can see there are a series of tests there, of tasks that allow us to generate a cell line to produce this recombinant monoclonal antibody. That culminates in MCB manufacture and testing, or master cell bank manufacture. That is the first GMP activity. By GMP, we mean activity that is part of manufacture of clinical material. And so the cell line development results in master cell bank manufacture and testing. The critical path on this slide, by the way, is along the dashed line. So keep your eye on that. Cell line development leads into process formulation and analytical development. So once we have a cell line, we need to develop a process by which that cell line will be turned into a product. That manufacturing process involves cell culture process and a purification process. We call that upstream and downstream. In addition, the drug product needs to be formulated in a way that it is stable, shelf stable, and can be stored. And also we need to characterize the molecule and release it so that we know what it is, what its characteristics are to ensure that it's quality. So that's what happens during that process formulation and analytical development stage. Based on these results, we make material for toxicology, that's in green, and use that material to do the toxicology studies and generate the results. In the meantime, we are making clinical material, and that's cGMP DS testing and DP testing. So that's drug substance and drug product. That is the large-scale production that will result in the clinical material that will be used for testing in patients. Some supportive data are required, and that's the viral clearance study in purple. That's a safety study that's required of in mammalian, in manufacturing processes that use mammalian cells. And then the drug product stability is required in order to verify that the formulation that we've designed does in fact result in a product that is shelf stable. So this is the 12-month timeline. And the question that Dar and I want to present tonight to you is, how can we move faster than 12 months? We think that there are 4 key elements to this. First, to adopt the latest technological advances. And what we mean by this, and we'll show you here in a minute, is this involves highly productive cell lines and the use of large bioreactors using single-use technology. By doing this, it enables the production of thousands of doses from a single batch of about 5 or more kilograms. Second is a focus on a platform approach. Now, this is possible with antibodies, and all of us who work in this field understand and are familiar with the platform approach that is so widely used. Most of these products are one particular isotype and one subclass, and that's human IgG1, expressed in one particular type of mammalian cell, and that's Chinese hamster ovary cells. In this circumstance, the safety and quality risks are low. Why? Because more than 50 of these types of situations have been commercialized, human IgG1s expressed in CHO cells. And so we have a substantial platform knowledge and a high level of experience and facilities for cGMP production of these types of products. We do, having said all this, need to be willing to accept a higher business risk. And we'll show you where this is needed in the subsequent slides. We have to consider the caveats, but with these caveats and with this higher business risk, we will not accept increased risk to patients in the first clinical trials. So this involves a determination of the risk benefit of making any moves to accelerate this timeline. And then the last point that we want to raise is that, yes, these are changes and improvements and efficiencies that we want to consider now in this pandemic circumstance. But as we go through these, we want you to think about why not after a pandemic? Which of these could actually be implemented once demonstrated to be safe and effective, or to be able to produce safe and effective material? Why can't it be applied after a pandemic? So let's talk first about faster cell lines. And each one of these slides where we dive into the details on these different task bars has the timeline graphic reproduced on the upper right of the slide. So you can see in this graphic which part of the timeline we're actually referring to. Under faster cell lines, the cell line development stage of work, targeted integration of antibody expression vectors is an important technological innovation that's come on the scene. We get more consistent expression and integration of low copy numbers in highly active transcriptional hotspots. This results in more high expressing clones that reduces the time for screening cell pools or clones, and the potential time savings of using targeted integration is about a month. Second, clone screening. Here we can incorporate imaging, which eliminates the requirement for a second round of cloning. The standard method of single cell cloning prior to the availability of imaging required 2 rounds of cloning by limiting dilution in order to ensure monoclonality of the cell line by a statistical means. But the advent of cloning a few years ago, the advent of imaging a few years ago made it possible to do just 1 round of cloning where monoclonality could be confirmed using the imaging method. And so incorporating imaging eliminates that requirement for 2 rounds. Second, the use of AMBER-15 versus 5-liter bioreactors eases the evaluation of many clones or conditions. And so if you want to evaluate 24 clones or conditions, and you want to use 5-liter bioreactors to do that, that's going to take an entire laboratory and many staff in order to conduct that experiment. In an AMBER-15, you could set that up in a hood with 1 technician and get those results much easier. The potential time savings here is as much as a month. And so you take these 2 points under faster cell lines, and the combined time savings, in our view, is 2 months with a relatively low risk. What about process development? How do we do this faster? So a supply run for downstream purification, formulation, and analytical development is needed. We need material in order to do the DSP. And so that can be supplied by a large-scale transient expression run. By large scale, it could be as large as 100 liters. This has become possible in the last several years. And this could be done earlier than stably transfected pool. It is more work than from a stable transfected pool, but it can be available 1 month earlier. So it saves us some time there. The use of a platform process. I mentioned this in a previous slide. What do we mean by a platform process? Well, it means that we restrict the selection of raw materials to those already in inventory in a CGMP warehouse. And the platform applies to all unit operations, so that there is no optimization of the various unit operations that would be considered here. It is what it is. It works for human monoclonal IgG1s expressed from CHO cells. It's not going to be the best process that anybody's ever developed, but it's going to be good enough. And so if we're looking for fast, then we have to accept good enough. The one step where variability is inherent is in the downstream polishing step. That step, as you know, is variable. That's usually cation exchange chromatography. It's going to vary with the particular characteristics of the antibody, PI in particular. And so this is a risk. So this step may require some optimization, but we would hope not very much. Because of the use of material from transient culture, the unit operations should be able to be defined before final clone selection. And so what this means, we think, is that whereas the current platform or the current timeline includes a demonstration batch, that would be a bench scale batch in which all the unit operations are combined. We would propose that one might consider no demonstration batch in this accelerated approach. Faster formulation development and stability. So due to the speed of CGMP production here, assessing the stability of the tox material might not be so useful. That may not be necessary in this accelerated mode. We might consider that antibody stability under accelerated and stress conditions could be compared with other monoclonal antibodies, and that product expiry dating would be extended by extrapolation then based on real-time data. Well, we said there could be no demonstration batch, and we want to take that one step further and say that perhaps we would do no evaluation of process performance at pilot scale. So this, the risk is increasing here. We're talking about rapid transfer to CGMP production scale following completion of cell line development. Now, one caveat here is that testing for bioburden and endotoxin are needed, and so we must allow time for this. We can't go into CGMP production without those tests. So whatever it is, whatever cell line is going into GMP production has to be tested for bioburden and endotoxin. But the platform process enables rapid scale up. So you take all of this together, and while it might work and it might work well, there is a high business risk. There's a lack of process confirmation that increases chances that these operations will not scale well, and that process performance and product quality will vary from the target. That's the risk that we take in considering taking these kinds of acceleration measures. Faster drug product. Oh, Dar, why don't you take
Oh,
over.
you were on a roll. I was going to let you go. But yeah, naturally from the manufacturer, we talk about faster drug product. And on this slide, the key assumptions here are that we need relatively large doses, so greater than 5 grams for these infectious disease indications. And these routes of administration, or IM, intramuscular, or intravenous injection, infusion, are the paths. This is technically your target product profile, your QTPP, if you will. These products are liquid, they're stored cold or frozen, they're between 50 to 75 megs per mil in terms of concentration. And you would only use formulations that are proven in excipients that are excipients that have been used in other previously approved products or studied in clinical trials. They're on the inactive list. They're generally regarded as safe. They're fairly well known in intravenous or solution type manufacturing. And the container closure systems would be those standard container closure components from, West and Schott and others that are tried and true. And while long-term stability wouldn't be required, we probably still need to generate some stability at the appropriate time. We're suggesting that we should still do it. We should do it longer because it's a batch probably. You keep it going for longer stability. The cost is low, but the benefit is high to just keep that program going for that drug product. Next slide. We talk about faster toxicology. What I didn't talk specifically about both the CTAP and the EUA processes is FDA relying really on animal models and animal safety, both in vitro and vivo. So it could technically remove some of the known pivotal tox studies that are done, typically from the critical path. We know that companies had to have done this successfully because of the time that we're seeing. And in this case, what Steve was talking about earlier about tox studies using material from transfected pools or the mini pools. And in this case, also, this accelerates those tox studies such as AIMS or tissue cross-reactivity, or generating those at least in vivo efficacy data in models or safety data in appropriate species that would help with accelerating that program further. Faster GMP production. For the people that have a quality background, regulatory background, you're probably twitching in your seat. But proposal to use the MCB while your testing is ongoing. So prior to having your full panel of testing, there is a business risk that you're using unreleased MCB in a GMP facility. And we say that we're focusing more on contamination. When Steve talked about the bioburden and the endotoxin, those aren't the kinds of things you want to transfer into a GMP facility or a CDMO. But in any case, the MCB testing would be complete before you distributed any product. In this case, you would derive the seed for the GMP production, as Steve was indicating, the expansion of the selected clone during the MCB and manufacture, so there would be no RCB established. It saves about 1 to 2 weeks, and in this time frame, that's a lot of time. And the GMP production would be at the smaller scale. So it'd be at 200 liters, you'd leave scale up for later. You have a shorter inoculum train, and you have lower risk to that particular 2,000 liter batch because you don't have to scale up and you don't have a pilot batch. Next slide. So in the case of operations, in terms of GMP production analytics, key is co-location of drug substance and drug product productions. It makes sense, right? More efficient operation, everything's in one location. You don't have to ship drug substance between sites. With your fill finish, you use disposable fill lines, you use robotic operations and isolators. We know that there are some CDMOs doing that right now and getting ready to put drug on the market. And this 2,000 liter batch would have 3 to 4 gram lead titer, and that would yield 300 to 500 1 gram doses.
200 liter batch.
I'm sorry, I said it again, didn't I? Drug product release testing. So we would qualify the platform on the analytics for the IgG1 monoclonal antibody. As Steve said, if the operations are platformed, qualifications should be equally platformed. The ELISA could be qualified and performed as conformity during the IND review, meaning you could qualify it, you could confirm it while the IND review process is ongoing. And the resupply batch could be further scaled. So you see here on the right-hand side, this is a 200-liter bioreactor, this cell line in that. But to scale up, you would go 10 to 20 times that. And you can see that you could do it by multiple manufacturers in these 1 to 2 liter production bioreactors. And with the downstream process and formulation testing. So leave scale up for later. Next slide, I think it might be, okay. So quality, faster quality. A lot of people don't think that's possible, but in this case, you talk about conditional release of both the drug substance and drug product. But again, as Steve mentioned earlier too, we focus on those safety tests that are important to make sure you don't contaminate facilities and process and equipment. So sterility ID, endotoxin, you would do those before you perform further processing, and all the other release tests would be forthcoming. And you could complete those tests while process is ongoing, or even during shipping or labeling or even packaging. So you wouldn't stop sort of downstream processes or operations from happening to wait for release testing. And when Steve talks about the viral clearance study in terms of this platform, it enables modular claims of viral clearance. So you don't have to do the viral clearance studies for a product specific viral clearance activity. You all know these processes well: low pH, virus filtration, protein A, and AEX chromatography. I think, Steve, that's my last slide. Is that right? Oh, no. My favorite topic. So in all of this, as I said in the beginning, you could do all of this, but in terms of what FDA and I'm sure the EU and other bodies are doing is thinking about focused regulatory. My suggestion for folks on the call or others that want to use this kind of accelerated production or accelerated manufacturing along with clinical, etc., is get familiar with what's out there. As always, it's early contact with FDA. Use the pre-EUA activity process and discussions. The guidance is fairly clear on the requirements. In terms of the CTAP, these pre-IND meetings. And as always, with drug development, is clearly understand the science and the plan. And really, it's another opportunity for everyone to be thinking out of the box. Steve?
Okay. So taking all of the these measures together, these measures for acceleration, we're presenting this slide here, which puts the traditional timeline, this is the 12-month timeline, and compares it to this potential accelerated timeline. The outcome of the traditional timeline is an IND. The endpoint of the accelerated timeline is a CTAP submission. And so you can see that cell line development, there is vectors, transfect, select, and clone, screen, and the rest of the stages are similarly compressed. I think the one point I want to make here again is follow the dashed line. You can see the path of the accelerated, the critical path for this accelerated program. Doesn't involve tox. One of the key features here is that the tox study is off the critical path, so that we are putting GMP production there on the critical path. The other is, as Dar pointed out, is that there are certain tasks that take place after filing. That's release and stability data, which need to be available prior to dosing. So those are key features of the accelerated critical path. I also want to point out in this slide that much of this material you'll find in Brian Kelly's article. That's one of our pre-reads. That's the Nature Biotechnology article in May of this year. So what's new here? We are primarily what's new is that we're integrating innovative CMC methods with a focus on speed. Targeted integration, a single round of cloning with imaging, use of the Amber 15, producing a development supply run from large-scale transient culture, a tox run from a cell pool, and no RCB, no research cell bank. So the growth of the production cell line is concurrent with MCB manufacture. Add to that abbreviated tox studies and package it all together by a willingness to accept higher business risk. All of that is what makes this accelerated timeline potentially happen. So I want to shift now in the last couple of minutes to covering the leading new monoclonal antibody therapeutics. All of these are currently in clinical testing, and so all of them in some form have incorporated an accelerated timeline, because we know they didn't take 12 months to get to into clinical testing. They took substantially less than that, and you'll see some of the details of some of these programs. We don't exactly know. We don't work with these companies. And so we don't exactly know what each of these sponsors have done, what shortcuts they've taken to get to this. But we know that I think it's reasonable to assume that probably several of the features of acceleration that we've covered have been done by these various companies. So let's talk about Lilly first. That's LY-CoV555. This is a monoclonal antibody. The molecular target, as we've discussed, is the spike glycoprotein of the SARS-CoV-2 virus, which, just to remind you, blocks viral attachment and entry into human cells. The sponsor here is Eli Lilly in collaboration with AbCellera, which is a company, a small company in Vancouver. Clinical data on this product is actually expected in the fourth quarter of this year. This is the leading antibody program as far as we can tell. The status of the program, all of these are genetically engineered antibodies that are recovered derived from the blood of a recovered patient. Just to underscore the point, the Lilly product required less than 3 months from screening of antibodies to first in human clinical trials. 3 months, and we're talking about 6 months. So you can see that these guys have done some very creative arranging of the various tasks required for development. Their phase 1 trial is completed, and they're currently moving into phase 3. The Regeneron cocktail, so that's REGN-CoV2. This is a cocktail of 2 monoclonal antibodies, also directed against the spike protein, but Regeneron has taken the approach that 2 is better than 1, and these 2 antibodies, while they both recognize the spike protein, they recognize different epitopes. So going back to what I told you before about trying to gum up the works of the lock, covering 2 epitopes means that you can be more efficient at gumming up the entire lock so that the key can't get in versus 1 antibody. This is sponsored by Regeneron. They have just recently inked a manufacturing agreement with Roche to expand their capacity, going back to Dar's slide on expanding from one 200-liter reactor to multiple 2,000-liter reactors. That's what's happened here. This is also genetically engineered from a recovered patient. Phase 1 trial is also completed, and they're currently in phase 3. GSK and Vir Biotechnology. This is Vir 7831, a single monoclonal antibody against the spike protein, genetically engineered from a recovered patient. And this one is modified genetically for increased stability in vivo. So it has a longer half-life. That increased stability might allow it to be stable in blood in a patient for up to 6 months, which would be great. GSK expects to move into phase 2/3 this month in August. And CTP59, this is sponsored by Celltrion in South Korea. Again, monoclonal antibody directed against the spike protein, and it neutralizes a virulent strain, a variant of SARS-CoV-2 D614G. Clinical phase 1 began in July in Korea for this product. So it's slightly behind the others. And then the last one is this product, a monoclonal antibody cocktail from AstraZeneca in collaboration with Vanderbilt in Nashville and DARPA. It's a cocktail of 2 antibodies, and like the Regeneron product, different epitopes of the spike protein. They expect to enter clinical trials this month. So in summary then, I just want to highlight what we've talked about for the last hour or so. The pre-pandemic monoclonal antibody CMC development timeline is 12 months from a product development decision to IND filing. Factors from several areas can be combined to shorten this timeline. The pandemic monoclonal CMC timeline could be reduced to 6 months. The CTAP pathway involves frequent dialogue and collaboration with FDA. And in our view, the CMC for these various products should be ready when a COVID-19 therapeutic antibody is authorized under one of these programs. With that, I'd like to open the webinar to your questions. So please let us know if there's anything that we can clarify.
Steve, while Kelly's doing that, I just wanted to add to that last slide is the therapeutics that are closer, the ones that are in phase 3. I'm sure that they've negotiated things with the FDA under the EUA process, the later stage ones, and I think that's part of the value of that process. The earlier ones, probably CTAP, we don't know, but I bet there's been a lot of negotiations and commitments, appropriately so, with the FDA in terms of requirements. But I think the focus is still always on the may be effective, right? I think that's really the kingpin of some of this is if it shows some benefit, then they get some help, you know what I mean?
Right. So I mean, I think what we just saw from the Mayo Clinic's convalescent therapy this past weekend is a good example of that, where, you know, under non-pandemic circumstances, the data that were presented are probably not sufficient, but under these circumstances, they are.
Right.
We do have one question, Steve, and I'm not sure who it would be directed to, but Sangeeta asks, will you evaluate the stability of your cell line or not?
Thanks for the question, Sangeeta. Yes, that would need to be evaluated, but it would be off the critical path. You might have noticed in the diagram, the timeline diagram, that the critical path line shifted to tox before cell line stability. And so even in the 12-month timeline, the current state-of-the-art timeline, cell line stability is off the critical path.
Okay, the next question comes from Russ, and Russ asks, are there any CDMOs that are implementing this accelerated approach? He's thinking of companies like Lonza, KBI, WuXi, etc. Do any of them offer accelerated technologies as you described?
Yes, yes. Several of them offer and incorporate the innovative technologies, one or more of the innovative technologies that we've included here. What we don't see from any of them is an integrated a program that integrates all of these technologies together in a commercial offering. That has not happened, at least in any kind of a formalized way. And Brian Kelly's article has proposed a number of these that these measures be kind of bundled together in this way, and that's what we're highlighting here. But I mean, if you go, for example, Dar and I and several of us participated just today in a presentation by Lonza in which they featured a supply run using large-scale transient transfection in order to save time in drug development. So we're hearing about these different innovations on a regular basis by CDMOs. We just don't see them all put together like this.
Okay, thank you, Steve. Next question comes from Joy, and it's for Steve, and she wants to know, do you know of any products being made from clone pools prior to single cell cloning for MCB?
For MCB, an MCB generated from a pool? I do not. We are aware of tox studies that are conducted using pooled cells. But no, I'm not aware of an MCB that's been generated from a pool. That's not to say it doesn't exist. And if there's anybody on the call who works for a sponsor who has done this successfully and submitted on it, please raise your proverbial hand.
Next question comes from Robert, and he says, some have suggested first clinical material coming from stable pools prior to clone selection. Can you comment on that?
Kelly, repeat that, will you?
Sure. Some people have suggested first clinical material coming from stable pools prior to clone selection. Can you comment?
Yeah. I mean, that is kind of the same question, right? So what that would imply that the clinical material is made from a pool of cells. I've not seen that done. That would be another measure, another acceleration measure. Dar, are you aware of anybody who's done that?
No, I'm not. That's an interesting one, isn't it?
Yeah. So the issue there is that the agency would have to abandon its expectation of monoclonality of a cell line. I'm not sure that it's prepared to do that. But, I don't know.
Yeah. In this situation, they might, right? It's all data driven. The question would be when the pandemic or the emergency is over and these things are quote unquote undone, to make sure that we've been thinking about how to go back to bridge the data sets that don't result in a maybe effective outcome rather than is effective outcome that you would need for a full approval. And
so you think it might fly under the EUA CTAP scenario, but
yeah, under the threat, right? Where it's, as long as there are data, as you saw the 4 criteria, the risk benefit may be effective. The may be effective is animal data, cell, in vitro data, previous human experience is something that might be similar that's already in the market. I mean, there's lots of ways to compile that may be effective to get them to at least consider it.
Right.
And the thing I wanted to respond to Sangeeta is, so we're not advocating, I don't think, Steve, we're advocating you'll skip all these steps, like we know we'll need to plug some gaps then, right? So there'll be work to be done to prove we have a quality product, that we have a process that's scalable and controllable, right? All those steps that we kind of leaped over to get to this. We have to think about what would be required to get a full approval. because PAI inspection is still, right? They've held off on those. FDA is finding ways to do virtual or different ways to do inspections. So it's not we're not advocating that you would never do it. It's what you said. It's not critical path.
Right, right. I mean, I think it's really all about what's changing here is the critical path more than anything else. I think that's really another take-home message.
Well, that's currently all the questions that we have right now. Are there any others that you all would like to ask before we conclude the webinar? You can either put it up in the chat or the question and answer tab at the bottom.
I think Steve put it out that if folks have any other ideas that we missed or hadn't thought about or a reaction to it, that'd be great to know.
And in particular, if any of you are at Lilly or Regeneron or GSK and you would like to share any thoughts or experiences, please do that as well.
Well, we did get one additional question from Robert. He says some have suggested that GLP tox has come in parallel to clinical testing for anti-COVID antibodies. Your thoughts?
Yeah. Sure.
I think that's what we proposed.
Yep.
The tox is off the critical path.
Right. As long as there's enough animal safety data to support what the FDA views as may be effective. Right. So we propose tissue reactivity in vitro, right? We propose maybe aims for mutagenicity. You would do an animal model for efficacy, mice, rat, whatever makes sense in terms of an appropriate species. So you would minimize that quote unquote tox package to just what makes sense. Robert, did that answer? Raise your hand or say yes or something. Nod your head.
He raised his hand. He did raise his hand, so
okay. All right.
Thank you. Thank you. Well, if there aren't any other questions, you have any other things that you would like to say before I launch the poll, either Steve or Dar? We did get a couple of thank yous from folks telling us that it was a great presentation, loved it. Thank you both. Great to see you.
All right, well, great. As we can't see any of you, but we know you're there. So thank you very much for joining us tonight. And please do, if you want to view these slides, they'll be available on our website. And just once again, take a look at the pre-reads. I think you'll enjoy them.
Thank you, everyone. The